This video is covering glucose metabolism part 2. So in particular today we'll focus on the Krebs cycle and oxidative phosphorylation. My name's Professor Joanne Lind. The learning outcomes for today's video are to explain the processes involved in carbohydrate metabolism in a healthy individual. Now, as mentioned in the part 1 video, this is a large topic, so I've broken it down into 2 videos where today, as just mentioned, we'll be focusing on the Krebs cycle and the electron transport chain. So you can see here, this is my map of metabolism. So in the previous video, we started with carbohydrates and we looked at glycolysis. So we looked at how you could break carbohydrates down into glucose 6-phosphate, so glucose to glucose 6-phosphate, and then via glycolysis continue that all the way to pyruvate molecules. Today we're going to be looking at how we take that pyruvate and convert it into acetyl-CoA and then break it down further under aerobic conditions via the Krebs cycle and electron transport chain. And I should mention that the Krebs cycle is also referred to as the citric acid cycle or the tricarbolic acid cycle. So just to remind you what we learnt in the first video, glycolysis is the breakdown of glucose, and the sequence of reactions takes that 6-carbon molecule to produce 2 molecules of pyruvate. Now, under aerobic conditions, we can completely oxidize that to carbon dioxide and water. So if we've got oxygen present, we can go down that middle track, and that's what we'll be talking about today. As shown in the previous video, glycolysis occurs in the cytosol of the cell. The product of glycolysis is pyruvate, and we need to transport then pyruvate from the cytosol into the inner membrane of the mitochondria, so the matrix of the mitochondria. To do that, it must first pass the outer mitochondrial membrane, which we suspect occurs through a voltage-gated anion channel, and then it needs to be transported across the inner membrane by a translocation protein so that it is in its place where it can then be further broken down, which we're going to look at now. So as mentioned, we need to get it into the matrix of the mitochondrion, so the inner, through the inner membrane. And to do that, we use a transport protein. Now, once pyruvate is in the matrix of the mitochondria, we need to then break that down to acetyl-CoA. And we do that with an important enzyme called pyruvate dehydrogenase. Now, this enzyme is important because we use it to convert pyruvate to acetyl-CoA, which is a non-reversible reaction. So you cannot make pyruvate again from acetyl-CoA. Once you've made that conversion to acetyl-CoA, you've committed that it has become acetyl-CoA and whatever you can do with acetyl-CoA. So importantly, you cannot then make those back into any form of glucose. So it's committing your pyruvate to some form of metabolic process that prevents it from going back to glucose. Now, this reaction from pyruvate to acetyl-CoA via this enzyme pyruvate dehydrogenase is carefully regulated because you don't want to commit the pyruvate to acetyl-CoA if the cell or the body is not in a state to be able to utilize that acetyl-CoA. So importantly, acetyl-CoA inhibits that reaction. So anything that is the product of that reaction, if you've got too much of it, why would you need to make more? So that inhibits it. We also know that acetyl-CoA can enter the Krebs cycle, which I'll show you in a second, and NADH is produced by the Krebs cycle. So if you've got a lot of NADH, it will also inhibit this reaction. This reaction is activated, however, by the reverse of these. So if you've got the low energy NAD+, which you need to skill in a way, give it more energy to make NADH, then this will activate this reaction to make acetyl-CoA. And it's all to do with the balance, so it's the ratios of how much NAD+ to how much NADH do you have versus how much acetyl-CoA to how much CoA do you have in the cell as to whether this pyruvate dehydrogenase reaction occurs. So in this video, we're saying it does occur, so we're in a state where we want to completely metabolize pyruvate to carbon dioxide and water.
Let's take a look at the major steps of the citric acid cycle. In the first step of the cycle, a 2-carbon molecule and a 4-carbon molecule are combined to form a 6-carbon molecule. That 6-carbon molecule undergoes multiple biochemical changes during the cycle, and at the end, the original 4-carbon molecule is produced. Each time a carbon molecule loses 1 carbon, a carbon dioxide is released. So 2 carbon dioxides are formed during the conversion of the 6-carbon molecule back to the 4-carbon molecule. So where does the 2-carbon molecule come from to begin the citric acid cycle? This molecule, acetyl-CoA, is made from pyruvate. Pyruvate is a product of glycolysis, the other major ATP-producing cycle in the cell. Pyruvate is transported into the mitochondrial matrix, where it is oxidized to acetyl-CoA by pyruvate dehydrogenase. This step produces 1 NADH and 1 carbon dioxide for each of the 2 pyruvate molecules made from glucose. The acetyl-CoAs are the starting point for the citric acid cycle. In the citric acid cycle, the acetyl group from acetyl-CoA is transferred to oxaloacetate to form citrate. 4 different enzymatic reactions then lead to the formation of succinate. These steps produce 2 carbon dioxides, 2 NADHs, and 1 ATP. Succinate is then recycled back to oxaloacetate through 3 more reactions. These steps produce FADH2 and 1 more NADH. All of the energy molecules made in the citric acid cycle, along with those produced by glycolysis, are essential for fully functioning cells, as errors in these pathways can lead to life-threatening diseases.
So as that video just showed, it showed you what happens once you've produced acetyl-CoA and how it then enters the Krebs cycle and goes round to produce NADH and GTP and FADH2, which are all high energy forms. Now, it's a cycle because once you've gone through one round of the cycle, you're back to having the same compound as you started with, which in this case is you start with oxaloacetate, it combines with acetyl-CoA, and as you move around the cycle, you end up back with oxaloacetate. Now, oxaloacetate is a very important regulatory molecule because if it is not available, then you cannot enter the Krebs cycle. Now, a number of people get confused about when and where that Krebs cycle will or won't operate depending on the blood glucose levels. Now, you need to think about the liver as a separate control center to the rest of the body. So it's in the liver, if oxaloacetate is not available, then the liver will not undergo the Krebs cycle. Whereas if you are requiring energy in the extremities of your body, then oxaloacetate cannot be used for other things in those extremities, so it will be available to undergo the Krebs cycle. So let's have a recap of that cycle. So we said we had acetyl-CoA coming in, and importantly, we're producing a lot of energy in this cycle. So we're producing NADHs and GTP and FADH2. These are all things that we are passing hydrogens to, and in doing so, we're actually passing electrons and we're passing some energy to these molecules, which we'll then look at in a second how they are then utilized to create ATP.
At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate.
So as shown in that short little video, we saw how the NADH and the FADH2 pass their electrons across the complexes in that inner mitochondrial membrane. So we're actually inside the matrix, which in this image is shown as the orange section, and we're passing those electrons through the membrane complexes, and in doing so, the movement of those electrons is facilitated by protons, so hydrogen ions, being pumped across that inner mitochondrial membrane. So they are actually going from a low concentration of hydrogen ion to a high concentration of hydrogen ion. Now that's very important because once you set up a concentration gradient, the high concentration will want to move back to the low concentration. And so what we're about to look at is how we can utilize that concentration gradient to produce ATP from ATP synthase.
ATP is synthesized by ATP synthase, a large complex of membrane-bound protein. Here we see ATP synthase, along with other membrane-bound proteins. Notice the large difference in the number of hydrogen ions on the 2 sides of the membrane. This difference is a hydrogen ion, or proton, concentration gradient. The energy associated with this gradient is used to synthesize ATP from ADP and PI. This occurs at the ATP synthase complex. 1 hydrogen ion enters the ATP synthase complex from the intermembrane space, and a second hydrogen ion leaves it on the matrix space. The upper part of the ATP synthase complex rotates when a new hydrogen ion enters. Once 3 protons have entered the matrix space, there is enough energy in the ATP synthase complex to synthesize 1 ATP. In this way, the energy in the hydrogen ion gradient is used to make ATP. Now let's watch the process again. Notice how the proton enters the ATP synthase and exits into the matrix space. Once 3 more hydrogen ions have crossed the membrane, another molecule of ATP will be made. In this example, the hydrogen ion gradient is large enough to produce 6 ATP molecules. Please watch as the remaining ATP molecules are synthesized. The process has now completed, and the result is an equal number of protons on each side of the inner membrane. Without a gradient, there is no more energy available to make ATP. In biological systems, however, a gradient is always maintained. The mitochondrial hydrogen ion gradient is generated as electrons pass through 3 membrane complexes. That process can be seen in the mitochondrial electron transport chain animation.
So if we look at how much energy we can actually get from a round of the Krebs cycle, it's all to do with how much ATP we can produce per NADH that's produced during the Krebs cycle. So we know that NADH contributes 1 pair of electrons to the electron transport chain. And we know that complex 1 pumps 4 protons per pair of electrons across that inner mitochondrial membrane to establish that hydrogen gradient. Complex 3 similarly pumps 4 protons across, still with this same pair of electrons, and complex 4 pumps 2 protons across. So in all, we have pumped 10 protons per NADH across that inner mitochondrial membrane. Now, as you saw in the video, we require 3 protons moving down its concentration gradient, so that's from the intermembrane space back into the matrix, so from high concentration to low concentration. So 3 protons moving in that direction helps produce 1 ATP molecule, which you can roughly equate to say that if you have had from 1 NADH 10 hydrogens pumped across the membrane and 3 hydrogens pumped back for every 1 ATP, so that's 10 out, 3 back in, that's approximately 3 ATP that you can produce per molecule of NADH. Now, this is debated as to what other energy might be used in this system, so it might not necessarily equate to that, but it's a good start so you can see that NADH ultimately produces more ATP than a single NADH molecule. So 1 NADH is approximately equal to 3 ATPs. So the summary of this short video is that we've watched how pyruvate is converted to acetyl-CoA in a reaction that is not reversible. So once you've committed that reaction, you won't go back. Now, if the cells have available oxaloacetate, then that acetyl-CoA will enter the Krebs cycle, and that is the most energy productive form of metabolizing acetyl-CoA. We know that the Krebs cycle produces 3 NADH, 1 FADH2, and 1 ATP molecule which is equivalent of the GTP. And that happens per acetyl-CoA. Now, if you go back to your glycolysis, you'll remember that you produce 2 pyruvates per glucose molecule. So you've produced 2 acetyl-CoAs per glucose molecule. So you can double all those numbers if you wanted to work it out per glucose molecule. We also looked at the electron transport chain and saw that the chain takes electrons from NADH and pumps protons across that inner mitochondrial membrane to produce that hydrogen gradient. Same things happen with FADH2, which you saw in that short video. But FADH2 is different because it actually enters that electron transport chain at complex 2. So if there's a problem with complex 1 in somebody's metabolism, so if they have a genetic mutation that affects complex 1, FADH2 can still be used to produce energy because it bypasses complex 1, coming straight in at complex 2. Finally, we looked at ATP synthase, which uses 3 protons, which moves down its gradient, so it wants to travel, so it's already got the energy to move in that direction, and it passes that energy to combine ADP with inorganic phosphate to produce 1 ATP molecule and water. Now remember, this only occurs if oxygen is present, because at the end of this reaction in the electron transport chain, you must pass those electrons ultimately to oxygen to make a water molecule. If oxygen is not present, you cannot operate the Krebs cycle. So my contact details are on this slide here, and as with the other video, there are some revision animations if you'd like to go through them step by step if you need more explanation of these metabolic processes. And there's also a fun YouTube I put in there where someone's actually put glucose metabolism to a whole song. So they sing it, and if you're the type of person who remembers things orally by hearing sounds, then I recommend you listen to that YouTube video. So thank you.